So we're doing chapter 7.1, which looks at energy in living systems.
So let's start with redox reactions. So energy production within a cell involves many coordinated chemical pathways, and most of these pathways are combinations of oxidation or reduction reactions. These occur together and are collectively known as redox reactions. So in reduction, an electron is gained by a molecule, and it also gains potential energy. On the other hand, oxidation is the reverse. It's when a molecule loses an electron and also loses potential energy. In oxidation, the electron isn't just lost, it's transferred from one molecule to another. And the use of electrons is the fact that you can transfer energy in small increments. So, electron carriers. So electron carriers, their job is to shuttle electrons between compounds, and they're easily reduced or oxidized. So important carriers are often derived from B vitamin groups and nucleotides. Two examples of which are NAD, or nicotinamide adenine dinucleotide, and FAD, flavin adenine dinucleotide. So NAD is derived from vitamin B, or aka niacin, and its most important use is gaining 2 electrons and 1 proton to become NADH. So the way this works is when electrons are added to a compound, they're reduced. So a compound that reduces another is called a reducing agent. So in the equation shown, RH is a reducing agent and NAD plus is reduced to NADH. When electrons are removed from a compound, it's oxidized. So a compound that oxidizes another is called an oxidizing agent. So in the equation shown, NAD plus is an oxidizing agent and RH is oxidized to R. So another example of a shuttle electron or electron carrier is FAD, derived from vitamin B2, or riboflavin. So NAD and FAD are extensively used in electron extraction from sugars, and NADP, which is another example of is actually NAD used to carry phosphates, plays an important role in anabolic reaction photosynthesis.
A living cell can't store significant amounts of free energy. Excess free energy would result in an increase of heat in the cell, which would result in excessive thermal motion that could damage and then destroy the cell. Instead, a cell must be able to handle energy in a way that enables the cell to store the energy safely and release it for use only as needed. Living cells do this by using adenosine triphosphate, or ATP. When ATP is broken down, usually by the removal of its terminal phosphate group, energy is released. The energy is used to do work by the cell, usually by the released phosphate binding to another molecule, which activates it. For example, in the mechanical work of muscle contraction, ATP supplies the energy to move the contractile muscle proteins. At the heart of ATP is a molecule of adenosine monophosphate, or AMP, which is composed of an adenine molecule bound to a ribose molecule and to a single phosphate group. Ribose is a 5-carbon sugar found in RNA, and AMP is one of the nucleotides in RNA. The addition of a second phosphate group to this core molecule results in the formation of adenosine diphosphate, or ADP, and the addition of a third phosphate group forms adenosine triphosphate, or ATP. Phosphate groups are negatively charged and thus repel one another when they are arranged in series, as they are in ADP and ATP. This repulsion makes ADP and ATP molecules inherently unstable. The release of one or two of the phosphate groups from ATP, a process called dephosphorylation, releases energy. Hydrolysis is a process of breaking complex macromolecules apart. During hydrolysis, water is split and the resulting hydrogen atom and a hydroxyl group are added to the larger molecule. The hydrolysis of ATP produces ADP, together with an inorganic phosphate ion and the release of free energy. To carry out life processes, ATP is continuously broken down into ADP, and ADP is continuously regenerated back into ATP by the reattachment of a third phosphate group. Water, which was broken down into its hydrogen atom and hydroxyl group during ATP hydrolysis, is regenerated when a third phosphate is added to the ADP molecule, reforming ATP. Energy must be infused into the system to accomplish this process of ATP regeneration. This energy comes from the metabolism of glucose.
The process of adding a phosphate to a protein is known as phosphorylation. In phosphorylation with ATP, ATP is the substrate and ADP is a product. In this process, ATP forms an intermediate complex with the substrate and enzyme in the reaction. This intermediate structure allows the ATP to transfer its third phosphate group to the substrate. When the intermediate complex breaks apart, the energy is used to modify the substrate and convert it into a product of the reaction. The ADP molecule and a free phosphate ion are released back into the medium and are available for recycling through cell metabolism. ATP is generated through two mechanisms during the breakdown of glucose. In the first mechanism, known as substrate-level phosphorylation, a phosphate group is removed from an intermediate reactant in the catabolic pathway, and the free energy of the reaction is used to add the third phosphate to an available ADP molecule, producing ATP. This is the most direct method of phosphorylation. The next mechanism is known as oxidative phosphorylation. 90% of the ATP generated during glucose catabolism comes from chemoosmosis, a process of ATP production in cellular metabolism. This takes place in the mitochondria of eukaryotic cells or the plasma membrane of prokaryotic cells. The process of using chemoosmosis in the production of ATP is called oxidative phosphorylation because of the involvement of oxygen in the process.